Okay, last time all the way through. So we begin the process of digestion up in the mouth. The mouth has a stratified squamous epithelium. This is also where you're going to find salivary glands. We ingest food into the mouth. Our very first step in our digestive processes, ingestion in the mouth. Propulsion by way of the tongue. We call this deglutition, and this is really just a fancy word for swallowing. So the tongue is going to compact the food that we've eaten. It's going to push it back towards the back of the mouth and into the pharynx. We have mechanical digestion, and this is accomplished by your teeth in the form of chewing, where we're really just crushing up the food that we've eaten into what's now called a bolus. And we will begin chemical digestion by the salivary glands. So the salivary glands will contain, or will make two enzymes, salivary amylase, which is going to begin digestion of starches in the mouth, which are long-chain carbohydrates, as well as we're going to begin digestion of fats with lingual lipase. We then move down into the esophagus, where we have a stratified squamous epithelium. Only thing happening here is propulsion, no absorption or secretion. The specific name for movement in the esophagus is called peristalsis, and it's just a rhythmic contraction that will move the now called a bolus down from the mouth to the next structure. So our lower esophageal sphincter will open, allowing the bolus of food to now enter the stomach. In our stomach, we've got a simple columnar epithelium, and this simple columnar epithelium is organized into gastric pits and gastric glands. Our gastric pits will contain mucous neck cells as well as surface mucus cells. So in the pits, it's all about mucus, and this mucus is going to create a protective barrier that will keep the mucosa of the stomach from being eaten by the contents inside of the stomach lumen, i.e. hydrochloric acid. All right, within our gastric glands, we have parietal cells, and our parietal cells will make hydrochloric acid which has a pH of 2, which is going to serve two functions. A, it's going to kill off any bad, dangerous pathogens that may be trying to enter the body, so it's going to be very protective in that way. But two, hydrochloric acid is going to denature our proteins, and this is going to allow us to more easily digest the proteins. We're also going to make, parietal cells will also make intrinsic factor, which is important in the absorption of B12, and B12 is important in our blood cells. It gives us energy, but it's very important in formation of our red blood cells and their oxygen carrying capacity. So if you actually don't have intrinsic factor and you can't absorb B12, then you can't live. You would die. It's called pernicious anemia. We also have chief cells, which are going to make pepsinogen, which is ultimately going to digest proteins once it's activated. Pepsinogen is the inactive form of pepsin, and gastric lipase. Enteroendocrine cells are our third cell type, and they're going to make a hormone called gastrin, and gastrin is just going to allow for the release of all of these other things. So gastrin will be released when there's chyme in the stomach. This bolus of food, once it passed through the esophagus and it enters the stomach, it's now called chyme. So once there's chyme in the stomach, gastrin is going to be released. Gastrin then causes the release of the components of the parietal cell and the chief cell. And so within the stomach, we've kind of combined propulsion and mechanical digestion in the form of the churning of the stomach. So that's kind of twofold. It's going to break things up a little bit, but also move things along. We also are going to accomplish chemical digestion in the stomach by way of gastric lipase, which will digest fats, and pepsinogen. Once it's secreted by our chief cells into the lumen of the stomach, hydrochloric acid, which is also in the lumen, will activate this pepsinogen to pepsin, and pepsin will then begin to digest proteins. Moving over here to the small intestine. Our small intestine has a simple columnar epithelium, and that simple columnar epithelium is going to be organized so that it has maximum surface area. So one of the ways by which we maximize our surface area are the plicae circularis, which are circular folds. Another way by which we increase our surface area is the intestinal villi, and the third way is the microvilli. So the intestinal villi, microvilli, and plicae circularis are going to be organization, sort of modifications of our mucosa that increase our surface area for absorption. So the mucosa is organized into intestinal villi with microvilli, as well as intestinal crypts in the small intestine. So within these structures, we've got our enterocytes, which are absorptive. We've got goblet cells, which will secrete mucus and keep things moving. And we have enteroendocrine cells, which are going to release cholecystokinin, or CCK, and secretin. So the processes that occur in the small intestine, first are chemical digestion. So the small intestine is really not going to secrete anything that allows for chemical digestion. It's going to get its secretions from the liver and gallbladder and the pancreas. So cholecystokinin is going to trigger the release of bile coming from the liver and the gallbladder, which will serve to emulsify fats. This is not actually chemical digestion. What's happening is we are more mechanically digesting and preparing the fats for chemical digestion. From the pancreas, we are going to have pancreatic amylase, which will digest sugars, pancreatic lipase, which will digest fats, and 3 different pancreatic proteases, which will digest proteins. And so the 3 pancreatic proteases are trypsinogen, chymotrypsinogen, and procarboxypeptidase. Again, released in their pro or their inactive form because they would eat up the pancreas that made them, because our pancreas is made of proteins. So these will not actually be activated until they're in the lumen of the small intestine. So once they're in the lumen of the small intestine, we have an enzyme released by our enterocytes called enteropeptidase, which will activate trypsinogen to trypsin. Trypsin will then activate chymotrypsinogen to chymotrypsin, and trypsin will also activate procarboxypeptidase to carboxypeptidase. All 3 of these red enzymes are going to digest proteins. And so we have a lot of our digestion really happening in the small intestine, specifically the duodenum. The very last steps of our digestion, of our chemical digestion, will be accomplished by important enzymes made by bacteria found in the microvilli. So these are called our brush border enzymes, and they're going to do the last steps of digestion, specifically of proteins and carbohydrates, not of fats. So the other important digestive process occurring in the small intestine is absorption. We're going to absorb most of our nutrients and most of our water here. So fats will be absorbed into a lymphatic structure called the central lacteal, which you can see drawn in green, which is in every single one of our intestinal villi. Carbohydrates and proteins will be absorbed directly into the blood. And so some proteins will also go into the central lacteal, larger ones, and some smaller fats will go directly into the blood. But for the most part, fats into the central lacteal, carbs and proteins into the blood. All right, over back onto the right, we have our large intestine, and this is going to be a simple columnar epithelium as well. Here we only have crypts. We do not have intestinal villi. We have the same cell types. We have enterocytes. We've got lots and lots of goblet cells here, more so than in the small, because at this point, we've really absorbed most of our water, and so we need something to kind of keep everything moving throughout here. And so we need more goblet cells to secrete more mucus to keep things rolling. We have an important sort of structural modification, which is a very thick muscularis called the tenia coli, which is this longitudinal muscle that kind of runs the length, we'll draw it here, of our entire large intestine. And so the tenia coli are longitudinal thick muscle layers that are going to kind of squish up the large intestine. It's going to create these pockets called haustra. So this is a haustrum. And so these pockets are going to allow for the sort of propulsion that we'll see in the large intestine. So processes occurring in the large intestine, chemical digestion, really this is just going to be the brush border enzymes. So final steps of digestion of our proteins and of our carbs accomplished by the brush border enzymes, just found within the mucosa. We're going to absorb the last vestiges of nutrients and water, and the haustra allow for this sort of special propulsion called segmentation, which is just kind of a localized mixing. So instead of rhythmically moving things forward, we have very slow movement here because we're kind of squishing everything and mixing it all up to make sure we get every last bit of water and every nutrient. We also have an important reflex called the gastrocolic reflex. When chyme is in the stomach, it triggers this reflex that causes 3 to 4 mass movements in the colon, which is basically going to make room for what's coming. So it's going to move things along in the colon when there's food detected in the stomach so that your body can kind of make way for the food that's in the stomach to get to the large intestine. Lastly, the rectum and the anus, we have a stratified squamous epithelium because We need to be able to withstand friction here. In the rectum, we're really only storing feces. That's its only real purpose. And in the anus, it is our very last step of our digestive processes, which is defecation, the process by which we remove any and all wastes that are not usable by the body. So there you have it.